Dear students, today's topic is coupling and repulsion phases or cis-trans phases of linkage. Actually, coupling and repulsion, these phases were first described by Bateson and Puneet in the year of 1906, and they performed several crosses in Lithyrus odoratus, that is sweet pea. Let us examine the crosses. Okay, this is the first cross where they took two characters, that is they follow Mendel's dihybrid cross process. Okay, these characters are, one is color of the flower and another one is shape of the pollen grains. Here the factors are, or genes are, capital P for purple color and small pp for red color. Similarly, for long pollen grains it was capital L, and for round pollen grains LL. Have a look. These are the parent generation. Phenotype is purple long, red round. Genotype will be capital P, capital L, and for another plant it will be small p, small l. These are the gametes. It is F1 generation. All the plants are purple flowered due to the presence of capital P and long pollen grains due to the presence of capital L. Okay. This is the checkerboard for F2 generation. Okay. These are the plants involved in selfing process, that is plants from F1 generation. They self them and then they obtained F2 phenotypic ratio as well as genotypic ratio. Okay. According to Mendelian independent assortment, it is like this. Okay, and it will follow the 9:3:3:1 ratio. 9:3:3:1 ratio. This is the conventional process. Okay. Since here the number of characters are 2, that is the cross is purely dihybrid one. Okay, so according to that, the expected result will be like this. This is the expectation. Okay, the total number they found in F2 generation is 6952. And that is why the expected number will be, for purple long it will be 3911, for purple round 1303, for red long 1303, and for red round 435. Okay, but the actual observed number are quite different. Okay, have a look again the table. These are the genotype, phenotype, expected number, then expected percentile. What is the formula to calculate percentile? Expected number divided by total number of plants into 16. Since it is dihybrid cross, then you will get the percentile. Okay. Now have a look on the observed number. The observed number are 4, 8, 3, 1, 390, 393 and 1338 respectively, and the total number it is same, 6952. So it is a huge difference between the expectation and the observation. It means that it is a deviation of Mendel's independent assortment fashion. Okay. Let us check the phenotypic ratio of F2. Our expected phenotypic ratio is 9:3:3:1. That is 9:3:3:1. But here the case is resulted in other way. 4831 is to 390 is to 393 is to 1338 is equals to 11 is to 1 is to 1 is to 3. Our expectation was 9 is to 3 is to 3 is to 1, but it becomes, or it became 11 is to 1 is to 1 is to 3. That is, it is a quite difference, and again we may clearly say that Bateson and Panith's result was not maintaining Mendel's independent law, assortment law. That is, Mendel's independent assortment fashion was deviated in this case. Have a look on the parental phenotypic frequency. Here the parental character are purple long and red round. And look at the table. Number of purple long is 3911 and number of red round is 435. This is expected figure. Okay, this one is expected percentile. That is expected plant according to law of independent assortment. It will be 4346, but observed number is quite different. Total number of observed plant which have the parental phenotypic frequency was 6169. That is a difference of 1823 plants. And here the parental characters, observed parental frequency number was 4831 and 1338 respectively. Okay, that is, here the observed numbers are greater than the expected number. That is, E is smaller than O. And if we look on the phenotypic ratio, again it is expected ratio is 9:3:3:1, but it was appeared as 11:1:1:3. So these are the parental frequency. According to Mendelian principle, it will be 9 plus 1 equal to 10. But the actual observed one is 11 plus 3 equal to 14. Here again, O is greater than E. Okay. Have a look on the other frequency, that is recombinant frequency. Here the recombinant frequency is tabulated. Recombinant phenotypic frequency at F2 generation. This one is expected number and its percentile. Expected number will be 1303 plus 1303 equal to 2606. But it is again deviated. Here the observed number is quite less than the expected number, and the numbers are 390 and 393. That is purple round, this phenotypic character, and red round phenotypic characters, that is recombinant characters, were appeared as 390 and 393 respectively. Okay. So here E is greater than O. Have a look. Expectation is greater than observation. That is, observation is less than the or smaller than the expectation. So it is clear that this cross is not maintaining Mendelian fashion. To confirm it, test cross is required. Okay, have a look on the conventional test cross result. This is a conventional test cross result of Mendelian principle. Here we follow the totally Mendelian principle. That is why I have taken the plant as P plant, and here the F1 plant is capital R, small r, capital Y, small y. That is round seeded yellow cotyledon P plant, and the recessive parent is wrinkled one, wrinkled seed with green cotyledon. So these are the gametes, and this is the result. Okay. Put the male gametes and then put the female gametes, then you fill up the Punnett board or checkerboard by yourself, and clearly it will be noticed that the phenotypic ratio of test cross is 1:1:1. That is 1:1:1:1. Okay, this is the conventional method, conventional result. Now have a look the test cross results performed by Bateson and Punnett. Is it maintaining 1:1:1:1 ratio? This one. So this one is F1 plant, purple long. These are its gametes, and it is the recessive parent, and this is the gamete. This one the checkerboard or Punnett square for F2 generation. So these are the genotypes and phenotypes respectively. Okay. You make the Punnett square, keep the gametes properly, and then fill up the board. Then you will get the genotype as in this manner, and the phenotype is purple long, purple round, red long, red round, etc. And the number of plants per 16 maintaining the phenotypic ratio is, for purple long it is 7, purple round 1, red long 1, red round 7. That is the phenotypic ratio. Here it is 7:1:1:7. Actual conventional ratio is 1:1:1:1, but it appeared as 7:1:1:7. Again, it is a deviation of Mendelian principle. Now they explained the cause behind it. Bateson and Punnett, they explained the deviation, and they said that since the dominant factors are from the same plant, in the parent generation we have seen that capital P and capital L are from the same plant or same parent, hence they tend to remain together generation after generation, and they prefer to enter in the gamete together, violating Mendelian principle. Okay. Again, I am quoting this. They said that since these dominant factors from the same plant or same parent, they try to be like this, P and L attached to each other for generation after generation, and they tend to present in the gametes together. That is why the parental phenotypic characters are greater than the recombinant one. In Mendelian principle, recombinant one was 3+3, but here the recombinant is quite less. Here the recombinant is only 1 + 1 = 2, and parental, in Mendelian principle, parental was 9 + 1 = 10. Here it is 11 + 3 = 14. The cause behind that, according to Bateson and Punnett, since the dominant factors are from the same plant, hence they tend to be present in the gametes generation after generation. They didn't prefer the independent assortment fashion, and they termed this togetherness of the dominant genes as coupling phase, okay, or cis phase. Clear? Okay. To make confirmation, they again performed another cross in which the dominant factors are present in different plants or different parents. Capital P from one parent, capital L from another parent. That is why these are the genotypes as well as the phenotype of the parent generation in the second cross. Here the genotype is capital P, small l, small l. That is why the gamete will be capital P and small l. And here the genotype is small p, capital L, capital L. That is why the gamete will be small p, capital L. So this is the F1 plant. F1 plant is having capital P, capital L together. That is why purple long. And to perform test cross, this one is the genotype of recessive parent. Okay, so this is the gene of gamete of the recessive parent. Okay, so these are the genotypes of F2 generation of this test cross. And according to this genotype, the phenotype are as like this: purple long, purple round, red long, red round. And the phenotypic frequency are 1, 7, 7, 1 respectively. Okay, look at the figure or look at the table or observe the F2 results. Okay, this one is parental character, one parental character, purple round 7. Then another parental character, red long, it is again 7. The conventional ratio was 1:1:1:1:1, but here it is 1:7:7:1. Again, Mendelian principle is violating. To explain this violation, Bateson and Panith, they said that since the dominant gametes are from the different plants, that is why these dominant genes repel each other and they didn't want to be together in the gamete. Okay, this property or this phenomenon was termed as repulsion phase or trans phase by Bateson and Panith. So from this discussion it is clear that if the dominant characters are from the same parent, then they will tend to be remain together generation after generation, and this is termed as coupling phase. And if the dominant characters are from different parents, then they will repel each other generation after generation, and Bateson and Panith termed them this phenomenon and as what? Repulsion phase or trans phase. Okay. Later on, Morgan in the year of 1910 postulated that these cis-trans phenomena are the two aspects of linkage. So when you will study linkage, then you will get cis phase and trans phase respectively. So prior to the study of linkage, you have to know what is cis phase and what is trans phase. That is why today we studied about these phases which were described by Bateson and Panith in 1906, performing crosses with Lithyrus odoratus. Thank you.